Hey y'all, and welcome back to Tech Fact Friday. When we left off on the history of biotechnology in Part 1, we had just finished our discussion on the very early efforts of biotech, like what was demonstrated with food fermentation by our ancient ancestors, up through vaccinations and genetics towards the end of the 1800s. In Part 2, we'll continue where we left off. So let's take a look at biotechnology in the early to mid-20th century. Between the years 1900 and 1910 is when much of the foundation of what we now know about genetics was founded. It was during this time that it was uncovered that traits passed through generations were carried by chromosomes, the X and Y sex chromosomes and their function were discovered, gene mutation and hereditary were explored, genotype, phenotype, and how those relate to biological organisms was defined, and it was found that certain genes could alter other genes. It's pretty fascinating that this was all 2 to 3 decades before the Great Depression. One of the last medicine-based discoveries in biotech before this shattering event was realized by Alexander Fleming in 1928. It was the use of penicillin in curing disease and infection. But before we jump ahead and pick up the thread again around World War II, we would be amiss if we didn't talk about how these genetic discoveries were quickly leveraged in a very problematic way. The revolutionary field of genetics was soon twisted by a form of racism justified by science called eugenics that declared some genes, and therefore certain humans, superior to others. This led to discrimination based on nationality, race, ethnicity, and an arbitrary measurement of mental fitness, culminating in the US passing the 1924 Immigration Act. Because the law was based on the 1890 census and not the 1921, this restricted immigration in a way that favored North and Western Europeans, essentially cutting off Asia, and severely limited South and Eastern European immigrants, which were some of the largest groups coming to America during that time. It contributed to very few Jewish refugees being able to flee to the US in World War II, and these eugenics-based restrictions remained until 1965. So, jumping ahead a little in our biotech timeline to 1938, physics and chemistry got involved with the research on inheritance and brought about molecular biology to view the physical structures on the macromolecular level by X-ray crystallography. Another jump to 1948 puts us at the production of the medication cortisone, an anti-inflammatory, and the first synthetically manufactured biotech product. Going back to DNA from the 1950s on, research on this accelerates exponentially. 1953 brings us the familiar double helix DNA structure, about 80 years after the discovery of DNA. It's amazing looking back on this from a modern viewpoint, because this well-known model is likely what you pictured when you heard DNA in the previous video. But the people who made the discovery of DNA had at best a vague notion of what it looked like. They just knew it was there and said, "Hey world, this thing exists." Anyway, the 1950s to 1970s were littered with DNA and genetic research, like gene mapping, genetic coding, the discovery of oncogenes, which aided in cancer research, Amy's testing to determine what chemicals can cause damage to DNA, and rDNA, or recombinant DNA, which is used to cut out certain parts of DNA that can then be combined with different DNA to create new genes. And on that science fiction sounding note, that's where we're going to end Part 2, because that's all we have time for today. In Part 3, we'll talk about modern biotechnology, discuss some current research, and maybe even look ahead to the future. Remember to share, like, and follow for more content like this. Until next time, it's time for an upgrade, a technical upgrade.